Zion, how are you?
I'm good. How are you?
Hi, Maria.
Hi.
Luke, good to see you. Happy Tuesday, everyone.
Happy Tuesday.
Tuesday's an interesting day, isn't it? Not halfway through, not beginning, not the end. It's just a day in the middle. All right, Abigail, good to see you as well. Anybody have any questions or anything they want to talk about? All right, let's begin. All right. So you guys are doing a great job overall. I sent out progress reports only to students who are not doing well this time. So if you didn't get a progress report from me, it means that I'm happy with the work that you're doing. So keep it up. All right. Now, last week, you guys learned about cell growth and you learned about cell division. But this is really important, not just for what you've already done, but for the information that we're going to cover in the next couple of weeks. So I want to do a really fast review. We're going to go quickly through cell growth and division. If you want more, stop me if there's anything that you don't understand. And then if there's time at the end, which there probably will be, we're going to talk about Punnett squares. So if you're staying with the due dates, you would have read about Punnett squares in the lesson that had a due date for yesterday on it. All right, we also are going to do a Kahoot to review mitosis right in the middle of class. So be ready with your either able to split your screen or do the on your phone. All right, so there's a couple of jokes for you. Today we're going to learn about mitosis. I need your undivided attention. And really, jokes about mitosis aren't funny, especially when you tell them twice. Twice, too. All right. You should never talk about mitosis at a party because it's a divisive topic. Isn't it interesting? Biology is the only subject in which division and multiplication mean the same thing. When we have cell division, we're actually multiplying the cells. Division, multiplication, yeah. And dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. Finally, this is a way that you can remember mitosis. Mitosis starts with, or rather mitosis, starts with one mic, and at the end you have two mics. All right, so we did learn about how cell size matters. A cell can only get to be so big. So take a look at these two dogs. You guys can see those, right? You see the picture? I shared it. Yeah, okay. So those dogs are the same size, but clearly the dog in the big room is in a room with more volume. The smaller room has less volume. So the bigger room also has more surface area, more wall space than the smaller room does. But the surface area to volume ratio is a lot less in the bigger room. So think of it this way. If it was your job, if that dog stayed in the middle, and it was your job to feed that dog and to carry away its waste products, it would be a lot easier to get to the dog in the smaller room than it was the bigger room, because the walls are a long way from the dog in the bigger room than they are in the small room. The same thing applies to a cell. A cell can only get to be big enough that its volume to surface area ratio is perfect for getting things in and then taking things out. So if the cell can't get to be too big, we need a solution. The solution is cell division. Make two daughter cells where you originally only had one. Now remember these terms. These terms are important. Chromatin, chromatid, chromosome. So this is chromatin, all this loose stuff that looks like spaghetti in a bowl. Now actually, it always looks like it's solid. We know now that chromatin can actually change its phase, and sometimes it acts more like a liquid than it does a solid. But in any case, it's unwound when it's not getting ready to divide. You don't have those X-shaped chromosomes when the cell's not getting ready to divide. So it does look a little bit like watery spaghetti. It is a wad of chromatin. So that's what this is right here, chromatin. Now, when it starts getting ready to divide, it begins to wrap itself up like this. The chromatin twists and turns around these proteins called histones. Then from the histones, it forms into these sticks called chromosomes. They're also called chromatids. This is where it gets a little bit confusing. This is a chromatid. This is a chromatid. This is a chromatid. But also this is called a chromosome. And the X shape is called a chromosome. So the two sister chromatids together are called a chromosome. One sister chromatid is called a chromosome. It gets a little bit confusing. It's a little bit like learning a foreign language. So just keep that in mind. Now, I want you to stop for just a minute and think about this amazing cell cycle. So this is happening in every cell in your body at every minute of every day for your entire life long. Now, in your body right now, every second, there are 2 million cells that are being replaced. 2 million, 2 million, 2 million, 2 million, every second, all day long. There are 86,400 seconds in a day. That means every day in the human body, there are 172.8 billion cells that go through this cell cycle. This is clearly pretty important. Now, these are all pictures of the cell cycle. So they all have the same thing that they're showing, but they show it in very different ways. This one has mitosis at the bottom. This one has mitosis at the top. This one doesn't even show the steps of mitosis. And this one details them here. And look at this one right here. This one has one we didn't even talk about, prometaphase, right in the middle. This is just an example that there's always another layer to anything that we learn. For our purposes, for our class, these four, prophase, metaphase, anaphase, and telophase, are enough. So those are the 4 parts of mitosis. So notice all of these pictures all have a longer interphase right here, and then they all have a mitosis phase. And we've got prophase, metaphase, anaphase, telophase. However you can remember it, just remember the order that they go in. Interphase is first, PMAT, IPMAT, IPMAT, whatever you need to know to remember. Now keep in mind, the most important thing of this whole cell cycle is DNA. The cell has to make an identical copy of its DNA so that we have at the end 2 copies that are identical to the one that came before. So when this cell splits into 2, each of those 2 new cells has a copy of that DNA. But did you guys ever stop to think about all the other things inside a cell? What about all the other organelles? We made a copy of the DNA, but I didn't read anywhere that we made a copy of the mitochondria or the endoplasmic reticulum or the Golgi apparatus. What about all them? What do you think? Cell doesn't make copies of them. Why not? They're not really important. Why aren't they, Luke?
I had a question.
Okay,
Are there cells within cells?
no. Nope, good question, but there are not. A cell is the smallest particle that makes up a living thing. Yeah, remember the cell theory, everything is made up of cells. Cells come from other cells. So there are never a cell inside another cell. Now, it gets a little bit tricky here because did we do I don't know if we did. Yeah, remember back when we were learning about, you guys will get a little bit of this when we talk about evolution. There was a time when one cell engulfed another cell. There was a time in history, way back when they think that life was evolving on planet Earth. There were cells that were able to perform photosynthesis, and they were engulfed by another cell. But they're not considered a cell anymore. Now they're considered chloroplasts. They can't function on their own. A cell can function on its own, but that's the theory anyway. And they think that what became mitochondria used to be a cell that was engulfed by another cell, but they're not now. So at one point in history, the answer to your question would have been yes. As of now, the answer is no. Okay, so what about those chloroplasts? What if a plant cell, what if when a cell divides, like right here, so we have prophase, metaphase, anaphase, telophase, now we have cytokinesis. What if this new cell doesn't have any mitochondria in it? Doesn't really matter. It's fine if it doesn't have any. The cell doesn't worry about those other organelles because it has the DNA that it needs in order to make anything that it needs. If you need more mitochondria, you need more Golgi bodies, they can just be made. All they have to do is make them, is duplicate them. So we have all this DNA that's in the nucleus. We have everything that we need to make it. This interphase at the very beginning, that interphase, the longer part of the cell cycle, right here, it's black in this picture right here. This is the period between cell divisions. So you can think of it as like the waiting room, but a lot of things happen in this waiting room. The first thing that happens is the G1, normal growth phase. That's where protein is being made and the cell is getting bigger. Then we have the S phase where DNA is being synthesized. That's what the S stands for. Then we have the G2, our second growth phase. And then finally, we go into mitosis. Now, take a look at these checkpoints. There's a checkpoint here, a checkpoint here. There's also one in metaphase. There's a checkpoint right there. At all these checkpoints, the cell is checking for errors. If it doesn't find any errors and everything is good, the cell continues on into the cell cycle. And mitosis right here, this is what the whole part of this is preparing for. And there's 4 parts: prophase, metaphase, anaphase, and telophase. We'll look quickly at all of them. So right here, this is the whole cell, this blue kind of globby thing there in the middle, that's the whole cell. So you've got mitochondria here, you've got the rough endoplasmic reticulum here. This cell is in prophase. You can tell because you can see the X-shaped chromosomes. It's the only time that you can see the X-shaped chromosomes all together inside the nucleus, which is beginning to break down. So you can also see these centrioles starting to form the spindle fibers. You can think of the centrioles as fishing poles and the spindle fibers as fishing wire. And they're going to grab a hold of something and pull it in like a fisherman would pull in a fish. So here's an animation that shows prophase. Here are our centrioles and our spindle fibers, X-shaped chromosomes, the nuclear membrane dissolved, it's gone. So you can see it going away here. As soon as those spindle fibers attach to those chromosomes, prophase is over. And now we move into metaphase. This is where the fishing wires reach out to the middle of the chromosomes and they pull the chromosomes into this flat plate in the center of the cell. Here you can see an animation of metaphase. The spindle fibers are moving those chromosomes into the middle of the cell. As soon as they're in the middle, it's over. Now in anaphase, the fishing wire shortens, kind of like the fisherman reeling in his catch, and those half of each chromosome is pulled into different halves of the cell. Notice also that this shape of the cell is changing. It's becoming like an oval shape here. That's because these microtubules on the other side here are pushing. They're pushing on that cell to turn it into an oval shape. And here you can see that happening. You see anaphase stands for apart. It's pulling those apart. And then once they're all the way over to the end, now we have telophase. Now in telophase, there's still one cell. It's still this one cell, but you can see it has 2 sets of DNA in it. This nuclear envelope is beginning to reform around the DNA, and the chromosomes are starting to unwind. They don't look like X's anymore. They're now looking again like that bowl of spaghetti, and the cell just keeps getting longer. So here's telophase happening. The nuclear membrane is going to begin to reform, and the chromosomes are separating into chromatin again. And then the last phase is cytokinesis. So at the same time that telophase is happening, cytokinesis is happening. And the cell pinches in at the center in an area called the cleavage furrow. And then these microfilaments contract, pinching the cell until finally we end up with 2 cells where we had only one. So you can see telophase happening there. And then here's the cleavage furrow forming. Cytokinesis happens, and you end up with 2 cells where you originally had just one. Whatever new of these organelles that are needed, the nucleus just gives the information to make them. So Let's look at the whole thing in 3D. So I want you to watch this in 3 dimensions. This is prophase. As soon as they're attached, prophase ends, metaphase lines them up in the middle, anaphase pulls them apart, and then telophase and cytokinesis happen at the same time, and we end up with 2 cells. But I want to turn it around so you can see them because it's all 3-dimensional. It's hard to see it when it's just a flat image. Prophase, line them up in the middle, that's metaphase. And then anaphase pulls them apart, telophase and cytokinesis. All right, now, because this is so important, I want to do a Kahoot to help you guys see how well you understand it. So for this Kahoot, because it's not a competition, I don't want you to use your real name. I just want you to put something in because I want this to be anonymous. This is for you so that you know whether or not you understand mitosis. If you don't score well on this, it just means that you need to go back to the lessons about mitosis and read them again, or contact me if you need more, if you need a different kind of answer, of support for that. All right, so do not use your real name. And here we go. www.kahoot.it. The game pin is 976588. Nine 76588. All right, need 3 more. 976588. All right, I'm going to go ahead and get started. I got one more person to join, but you can always put this code in after we get started. All right, here we go. There's only 14 questions in this, so it will go quickly. You have 10 seconds to answer most of them. There's a couple that you have 20. All right, is this true or false? That is true. Remember, we duplicate that DNA so that at the end we get 2 genetically identical. They have exactly the same DNA. So that is true. All right, question 2. You're going to put the 4 phases of mitosis in order. So click the first one first. We have interphase first. Then what are the 4 stages of mitosis? Prophase, metaphase, anaphase, telophase. Okay, nobody got that one right. All right, that's something you need to learn. Knowing those will help later, believe me.
I couldn't write it fast enough.
Oh, all right, I'll add more time next time. What do we call the period between cell division? The waiting room. That is interphase. Interphase. All right, question 4. Which phase of mitosis does this image show? Notice you have the nuclear envelope there, 1 set of X's. Nuclear envelope is breaking down. That is prophase. Prophase. So prophase, you can still see the nuclear envelope right there. It's dissolving, but you can still see it. And they're X shapes, but they're not in a line in the middle. They're kind of still scattered. All right, next question. In which phase do duplicated chromosomes become visible and a spindle begin to form? they're duplicated in the S phase of interphase. Then that first phase where you can start to see them and those spindles begin to form is prophase. Prophase. In which phase do chromosomes separate and move toward opposite ends of the cell? All right, everybody got that one. Anaphase, anaphase. Anaphase means apart. They're being pulled apart. All right, now you're going to type your answer. If a cell has 24 chromosomes, how many chromosomes will each daughter cell contain after mitosis? You can do the number or the word, either one. Correct answer is 24. 24. All right, so only two of you guys answered that one. That must have gone quickly. So remember, they're identical. They're genetically identical after mitosis. So if the parent cell had 24, the daughter cells will have 24. If the parent cell had 5, the daughter cells would have 5. All right, question 8. Which process completes cell division by splitting one cell into 2? That is cytokinesis. That's the very last stage. Completes means ends. It ends cell division. You started with one cell, they split off, and you have 2 cells. Which phase of mitosis does this image show? No nuclear envelope anymore. X-shaped chromosomes in the middle of the cell. Metaphase, metaphase, good. Few more questions. In what phase does the nuclear envelope reform around chromosomes and the nucleolus is visible? That is telophase. Telophase. Prophase is the beginning when the nuclear envelope breaks down. Telophase is where the nuclear envelope reforms. Which phase of mitosis does this image show? That is anaphase. They're being pulled apart. See, we just still got one cell, one set on, one individual chromosome on each side, no nuclear envelope reforming. They're being pulled apart. How is cytokinesis in plant cells different than in animal cells? Plant cells form a cell wall around the outside. Animal cells do not have a cell wall. Which phase of mitosis does this image show? Two nuclei. That's your hint. That is telophase. Telophase has 2 nuclei. And the last is a poll. How confident are you in your knowledge of mitosis? All right, not confident yet, need to study more. Yeah. All right, so you guys know how you did on that one. If you didn't score well on this, then you need to go back to the lessons and relearn the stages of mitosis and what they mean. This really will become important later. So whoever Flower is, well done. Flower got the highest score. All right, let's leave that one. And guys, I'm really sorry. I have to take like a 2-minute break. Okay, 2 minutes and then I'll be right back. I got something I got to emergency take care of right here. So I will be right back. Okay, that was less than two, I think less than one. Thank you for your patience. Sorry about that. All right, so now we are going to talk a little bit about Punnett squares. So last week, you guys read about this guy, Gregor Mendel. Remember, he is the scientist who first started researching about genetics. So we call him the father of modern genetics. Now we'll learn as we go through genetics that a lot of what Mendel believed was later proven to be wrong, but a lot of it was right. And what was really important about Mendel, not only did he get us started, before Mendel came along, people really didn't think very much about genetics, how you ended up looking like your ancestors. They didn't think about that. But Mendel did. So he got us started and he used really good lab procedures. And that was important. That was something scientists weren't really doing before him. He took meticulous notes, and that's really important for a scientist. Now, part of the reason that he was successful was because of his choice of plant. He used peas, and they're perfect for this kind of study. They have a lot of contrasting characteristics. So they have like round seeds or wrinkled seeds, purple flowers or white flowers. Easy to tell the difference between these two. They also have a pretty short life cycle. You don't have to wait very long between generations. They produce a lot of offspring. They can be handled pretty easily in a laboratory. Now think about all that and compare that with like people. You know, we don't have a short life cycle. We don't have lots of offspring. We're not very easy to handle in a lab. So peas were the perfect plant to do lots and lots and lots of studies with. Now, yesterday you started reading about Punnett squares if you're sticking with the due dates. Now I know Punnett squares can be a little bit intimidating. Once though you learn how to make one and use them, they are very useful and very easy. So we're going to spend the next half an hour now practicing using Punnett squares. So if you understood yesterday, great, this will be easy for you. If you didn't, or you haven't gotten there yet, then hopefully this will be a helpful review. So this is a Punnett square. It's just 4 squares with space on the outside like that. It is used to predict the possible genotypes for the offspring of 2 known parents. What do I mean by genotypes? Can anybody remember what a genotype is? I know we've had this term before.
Genetics.
Yep, it is the genes that a person inherits or an organism inherits. It is their genetics. Let's look at a couple of important terms that we need to understand. So one of them is genotype. We'll get to the opposite of that in a second, but let's look at this one. What do we call a gene or an allele, which is a form of a gene, whose trait always shows up when it's present? It's the bossy one right there. Always shows up when it's there. What do we call that allele?
Dominant?
Yep, that is dominant. So then what do we call the other one? The trait only shows up when the dominant allele is not there. What's the opposite of
Not
dominant?
secondary.
It is secondary, but that's not what we call it. We call it recessive. We've got a dominant trait and a recessive trait. So you can see them right here. A dominant trait dominates or covers up the other form of a trait. A recessive one is dominated or covered up by the dominant one. So we're always going to express our dominant trait as an uppercase letter. And then whatever the recessive trait is gets the lowercase letter of the dominant one. So for instance, if we're talking about flower color and purple is dominant, the dominant allele is going to be a big P, but the lowercase p is going to be the recessive allele. It's not going to be like W for white. All right, now a couple more important terms. We said the genotype is the genetic makeup of the organism. The phenotype is the observable form of the organism. So in this case, the genes here are the genotype, and what it looks like or is like is the phenotype. So in, using our examples here, phenotype is usually, it would be words that we would use to describe it. It's tall or short or green or wrinkled, where the genotype, we use the alleles that we have. So here again, the genotypes are at the top. It's the genes that you inherit. The phenotype is the physical appearance of something. All right, now that we know the terms, let's make a Punnett square. So when we make a Punnett square, we start with our box and we put our parents' alleles on the outside. So in this case, there would be a dominant and dominant on the outside there, recessive and recessive there. So to make the Punnett square, we simply drop the letters from the top down and carry the letters from the side over. So we're going to drop this B down here into both of these squares. We're going to drop this uppercase B down into both of these squares. Now that top is done. We've dropped the uppercase B, the dominant allele, down into both. We're going to carry over our lowercase to both of these, And these are the possible genotypes for the offspring of these two parents. All right, let's do another one here now. The parents' alleles go on the outside of the square. So if I drop this one right here down, I'm going to get this, dropping it into both boxes. Now I'm going to drop this one here down into both boxes. This one right here, I'm going to carry over from the left into both boxes. And now finally, I have this one. All right, you guys, anybody not understand how you make a Punnett square? How you calculate? All right, now let's see what we can do with it. All right, so parents' alleles go on the outside of the square. Let's say we're trying to find the possible outcomes, the possible genotypes of a cross between black and brown feather color in a particular bird. We're told that black is dominant over brown. So we know that black is going to be an uppercase B. That's going to be black feather. The lowercase b is going to be for brown feather. The parents we are given have the genotypes homozygous dominant and homozygous recessive. So now we've got to put them on the Punnett square. We're going to put the parents on the outside. That's this one right here. Right here. And now we have the homozygous recessive one on the outside right there. All right, now we're going to drop the letters down. So we're putting the big B's drop down, the little b's carry over. Little b, little b, little b, little b. All right, now the order doesn't necessarily matter. We could have done it like this, little b, big B, but a good rule is to put the uppercase one first, with the big B first. That's a good rule of thumb to follow. That's what they almost always do. All right, so taking a look at this Punnett square, let's evaluate it. What do the results show? If big B is the dominant allele for black feathers and little b is the recessive allele for brown feathers, we can make predictions about the outcome. So what do we see for our results? Somebody evaluate that for me. What is the likelihood of the offspring going to be? First of all, let's do their genotype. What percentage are going to have any particular genotype?
Is it 50-50% chance?
So look at the inside of the square. Are 50 of them you know, like, what do you see inside all four of those squares?
Could it be 25, 25, 25, or like 25% chance or no?
It's actually, they're all big B, little b. So they all have this genotype,
So it'd be 100%, right?
100%. Yeah. So that's the genotype, 100% big B, little b. That is our genotype. So we're going to have 100%. Let me put the genotype on first. The genotype 100% big B, little b. Since big B is dominant for black feathers and they all have at least one big B, we're also going to have 100% black feather color in these birds. Okay, does anybody not understand that? Everybody see that? Well, let's do another one here. If big T is the dominant allele for plant tallness and little t is the recessive allele for plant shortness, dropping down this T here, what's going to go right here?
Lowercase t.
Lowercase t from there. Yep. And how about right here? It's going to be an uppercase T. And then right there,
Lowercase.
lowercase t. Excellent. All right. So tell me what is going to be in this box right here.
Another uppercase t.
Another uppercase T because we come over from here. We have another uppercase T right there. All right. Great. What's going to go right here in this box?
Another uppercase t.
Yep. Another uppercase T and then the little t that came from there. How about this one here?
Lowercase t.
Lowercase t comes over from that side. And then finally in the last box, another lowercase t. All right, now we can look at our results. If big T is dominant for plant tallness, any of those that have at least one big T are going to be tall plants. So what percentage of the plants here are going to be tall?
75%.
75%. You got it. 25 for each square. Losing my, so that one has at least one, that one has at least one, that one has at least one. So 75% are going to be tall. 25% are going to be short. Now look at our genotype. The genotype are the letters themselves. What is our percentage of each? First of all, how many different genotypes do you see there? Say again, Luke, I didn't hear you.
4 genotypes.
Well, there's 4 total, but 2 of them are the same.
Oh, 2 same genotypes.
These 2 are the same, big T, little t. So we have 3 different genotypes here. We have the big T, the little t, and the big T, little t. So we have homozygous dominant, homozygous recessive, and heterozygous. So our genotype is going to be 25% big T, homozygous dominant, 50% heterozygous, and 25% homozygous recessive. All right, once again, stop me if you want more information. If you don't understand how I'm getting this.
I have a question.
Yes.
So, like, you know, this chart we're using, is
Yep.
it possible for people to like use this chart on humans rather than plants?
Yeah, absolutely. In fact, I have one here. Let's do that one next. So here we're going to use it to determine the likelihood of getting a male or female child whenever there's a fertilization. So we have a female here and we have a male here. Now the problem with male, female, with the X and the Y chromosomes is there's not one that's dominant over the other. They share dominance. They like are correspond with one another. So if you have one X and one Y chromosome, you're male. If you have 2 X's, you're female. Simple as that. So let's do the cross on this. What's going to go in this box right here?
X.
But you need 2.
2 X's.
2 X's. There you go. Just like that. What's gonna go in this box here?
2 X's.
2 X's. How about this one right here?
What?
XY, and in the last one?
The same thing, an X and a Y.
An X and a Y. So what are the odds of getting a female child?
50-50% chance.
50-50. 50% of getting female, 50% of getting male every single time. So every time a baby is conceived, there's a 50-50 chance of having male or having a female child. Yeah, and we can use this not just for X and Y chromosomes. We can use a Punnett square for all kinds of genetic information. And we do. Punnett squares are a very useful tool. So let's do one with a fruit fly. So this is a cross between a brown-eyed and a red-eyed fruit fly. Now, in this Drosophila melanogaster, which is the fancy name for fruit flies, red eye color is dominant. So we use the big R. Red is dominant over brown eye color. Now notice we don't use a little b for brown eyes. We use the lowercase of the dominant one. So red eye color is dominant over brown eye color. If the flies in the picture were crossed, what percent of their offspring would be expected to have brown eyes? So we do a Punnett square in order to figure that out. Let's put this parent right here. There's my little thing there. Let's put this parent right here on the top. So we're going to put the big R and the little r on the top. Now we're going to put this parent fly on the left. 2 little r's. What goes in this box number 1 right here?
A capital and a lowercase r.
Yep, very good. What goes in number 2?
2 lowercase r's.
You got it. What goes in number 3?
A big R and a little R.
Yep, exactly. And what goes in the last one, number 4?
Two little R's.
2 little r's, right. Now, we would call these heterozygous because there's one of each, one dominant and one recessive. So there are 2 that are heterozygous and there are 2 that are homozygous recessive. All right, now let's answer the question. Our question was how many of the offspring are likely to have brown eyes? What percentage do you think are likely to have brown eyes? Remember, brown
50%.
50%, exactly. In order to get brown eyes, you have to have 2 recessive alleles. So 50% of them are going to have both of those recessive alleles. 50% of them are likely to have the homozygous recessive. Now, just like with the one that we were looking at with this one here, this does not mean that if these 2 parents have 4 children, 2 of them are going to be female and 2 are going to be male. Every single time there's a 50-50 chance. So the same with the fruit flies here. If we look at this, every single time, every single offspring that they have, there's a 50% chance of it having brown eyes and a 50% chance of it having red eyes. Make sense? All right. We can also go backwards. If we know what the parents are, or what the offspring are, sorry, we can figure out the genotypes of the parents. So let's see how we do that. Let's take this B. This B came from somewhere. This has to be a big B. And so does this one right here. In order for this to be big B, these both have to be the dominant allele. So does this one right here, because this B came from here, but this one has to have come from here. Now, what does this one right here have to be?
Lowercase.
Lowercase. Has to be a lowercase b. Right there. In order to get this one and this one, that little b had to come from there. So which 2 are the genotypes of the parents? The first one there, A. Big B, or homozygous dominant and heterozygous, one of each. That is the correct answer there. All right, let's do this one now. Look at that and tell me if that's accurate.
No, it's
Why
not
not?
accurate.
Why not?
Because the first column isn't picking up the capital T,
Right,
and one of the
exactly.
parents is T, capital TT.
Yep, there should be an uppercase here and an uppercase here in order to make that accurate. Good. How about this one?
Accurate.
Yep, that is accurate. So what would we call all of the offspring? Heterozygous, homozygous dominant, or homozygous recessive?
Homozygous dominant.
Homozygous means both alleles are the same. So in this one, both alleles are not the same. We have a dominant one and a recessive one. So this is going to be called heterozygous. Heterozygous. All right, so which parent, the one on the top or the one on the left side, is homozygous dominant? Homozygous means both alleles are the same. Dominant means it shows up whenever it's there. Whenever its trait is, whenever there's at least one allele, it's going to show up. This is dominant. The uppercase is dominant, the lowercase is recessive. So the parent on the left is homozygous dominant. The parent on the top is homozygous recessive. All four offspring are going to be heterozygous. All right, does that make sense? All right, so let's fill in the boxes here. What goes in box number 1?
Capital R, lowercase r.
You got it. What goes in box number 2?
Same thing, capital R and lowercase r.
Very good. What goes in box number 3?
Lowercase r, lowercase r.
Yep, and number 4.
Lowercase r, lowercase r.
You got it. Now, what is the probability of the dominant trait showing up in the offspring? Luke?
Sorry, I had a question, but I could wait.
Okay, no, go ahead.
So I had a question like in regards to like human fertilization.
Yep.
Is it possible, like, say if you had like two parents from different ethnicities, if like a child could pick up only like one parent's ethnicity, like if they were to do a DNA test?
No, it is not possible.
Okay.
You're always going to have genes, you're going to have alleles from both parents. It is not possible to get all of any allele from one parent.
Okay.
Now there are some genetic mistakes that happen, some disorders, if you will, where you might inherit 2 alleles from one parent, but you still get the other one from the other. So in that case, you'd have 3, and that is an anomaly. That's a disease, a disorder, if you will.
Oh, okay.
Yeah, you're always going to get at least 1 from each parent, and it's really complicated. So we're going to learn about what's called meiosis in the next week. That's why learning about the stages of mitosis are so important, because knowing about meiosis will help you to understand that that's how you get that's how cell division happens when you're making egg and sperm. And then you get half of the chromosomes of your dad went into the sperm, half of the chromosomes of your mom went into the egg. When those then combine in fertilization, you get traits from both parents. Yeah, but there's also switching around of the DNA. It's really the nature goes to a lot of trouble to make sure that we're all unique, that we're all different from one another. Yeah, that's a very good question. All right, so the probability of having the dominant trait in the offspring here is 50-50, 50%, because 50% or half of our offspring are heterozygous. What are the other ones? What do we call these here? If the top 2 are heterozygous, what are the bottom 2 called? I'll give you a hint. They're homozygous something. Are they homozygous dominant or homozygous recessive?
Homozygous recessive.
They are homozygous recessive. In order to be homozygous dominant, you would have to have two of the dominant alleles, two big R's. All right, so which allele is dominant in this one? If I said circle a dominant allele, what would you circle?
Lowercase y?
That's recessive. We're always going to show the dominant allele with the uppercase letter. It's always going to be dominant. It's going to dominate. The big one's going to dominate over the little one. The dominant allele will be the uppercase Y. All right, so right here, we have heterozygous. Why do I call that heterozygous? Hetero means different. It's a prefix. When you see that in front of a word, it means different. So they have two different alleles. They're called heterozygous because they have one dominant and one recessive. What are these two here on the bottom? They're not heterozygous because they're not one of each. They're both the same. So what do we call these? They are homozygous recessive. Homozygous dominant would be two big Y's. Homozygous recessive is two little Y's. Heterozygous is one big and one little, one of each. Now, the dominant allele is always the uppercase right here. So here we have one homozygous dominant parent. Sorry. We have one heterozygous parent because it has one of each. What are these two going to be? The three options are heterozygous, homozygous dominant, homozygous recessive.
Homozygous dominant.
Very good. Homozygous dominant. What are these two?
Heterozygous.
Very good. Heterozygous, because there's one of each that is represented there. All right. What are the odds of getting an offspring that shows the dominant trait with this cross? What percentage of the offspring could be expected to have the dominant trait?
All of them, wouldn't it?
All of them. Yeah, 100%. What is the possibility of having the recessive trait show up?
50%.
Well, if they're all going to be dominant,
Would it be 20?
Oh, 0%.
yeah, it's going to be 0%.
Aw,
You got it. Yeah, there's a 0% chance. In order to have the recessive trait show up, we would have to have two of the lowercase y's, and we don't. All four of these have at least one. At least one. All right. Anybody have any questions? All right. Hopefully that helped if you haven't gotten to it yet, or helped to review if we did. Let me see here. Oh, one last thing I wanted to show you before we're done. So we've been doing what's called a monohybrid cross. You can also do dihybrid crosses. Now this looks a lot really intimidating, but I want to show you how easy it is to fill it out. A dihybrid cross studies two traits at the same time. So this would be like smooth yellow seeds. So this one right here is going to carry down to here. I want to show you how you fill this out. You drop the big one down, just like we did before, but we're dropping down two letters at a time. Now with this next one right here, we're going to drop down a big S and a little y in all four boxes. In the next one, we're going to drop down a little s and a big Y in all four boxes. And then the last one, we're going to drop down a little s and a little y in all four boxes. Now we do the same thing, carrying it over. For this one right here, right there, we're going to carry over a big S and a big Y. Then a big S and a little y, a little s and a big Y, and a little s and a little y. So when you look at this dihybrid cross, what are the odds of getting a homozygous recessive in both traits? There's 16 squares. This is the only one that shows homozygous recessive in both traits. So it would be a 1 in 16 chance of getting homozygous recessive for both traits. Yeah, you can actually do trihybrid crosses too. We won't ever do that. They're really confusing to do. All right, if you don't have any questions, you guys are free to go. Thanks for class today. I'll see you
Thanks,
next week.
Jay.
Bye, everybody.